Hello students, welcome. Now today we will start with the actual mechanism of transcription in prokaryotes. We are done with the basics in transcription. Now it will be easy for you to understand the mechanism of transcription. As you will be knowing that in your syllabus there is just transcription in prokaryotes. Prokaryotes know the organisms which don't have any well organized nucleus. Now, states of transcription. Transcription, it is synthesis of RNA from DNA. Its states are initiation of transcription, elongation, and termination. Also, it's important to remember that the site of transcription in prokaryotes is cytoplasm, as they lack a well-defined nucleus. Now, firstly, coming to initiation. As you remember that the important thing in this transcription is that we require a DNA molecule from which message is to be transferred to the RNA. And also you remember, as we have talked in the basics of this transcription, that the polymerase which catalyzes the formation of this polyribonucleotide of RNA, it is bacterial RNA polymerase, of which that is multi-subunit. There are alpha subunits, beta, beta dash, and omega, and then we are having sigma. This sigma, it keeps on associating and dissociating with the RNA polymerase depending upon its requirement, and is mainly required for the promoter recognition. And this enzyme, excluding the sigma subunit, it's called as coenzyme and is required for actual polymerization, for actual catalysis. Now, the first step in the initiation is that this bacterial RNA polymerase, along with the sigma subunit, now scans this RNA chain to find a promoter. And once it finds the promoter, it binds there. See this yellow, it's a promoter, and the RNA polymerase binds here. Well, binding to the RNA polymerase by the sigma subunit, it prevents the wasteful process of synthesizing all the RNA. It just synthesizes the RNA that's actually required. Now we are having RNA polymerase that has found its promoter with the sigma subunit bound to it, and we are having a DNA helix. Now, as you can see, the DNA duplex, it's still having the hydrogen bonds intact and is closed. So we say it's a closed complex. And another thing is it's called a binary complex. Why binary? Because there are two molecules of different nature, different chemical composition. One is DNA and another is enzyme. So we call it as closed binary complex. Closed because DNA is still in its intact form, and binary. Why? Because two molecules of different chemical nature are involved. One is the DNA and another is the enzyme. Now continuing with the initiation. When RNA polymerase binds to DNA, it covers about 70 to 80 base pairs. The closed complex is now turned into open complex by melting a short stretch of DNA within the sequence. Now what happens? This RNA polymerase, once it finds the promoter, it moves along these hydrogen bonds, breaks them, uses its helicase activity, which is more specifically, if you remember, in the beta subunit, and unwinds this DNA, creating a open structure which was still now closed. It now creates it open, and it unwinds about 12 to 14 base pairs of DNA, creating a bubble-like structure which we call as transcription. Here in this picture, if you are able to see that the RNA polymerase, it ventures through this DNA and breaks the hydrogen bonds, uses its helicase activity, breaks the hydrogen bonds, and as a matter of fact, a bubble-like structure is formed which we call as transcription bubble. Now, if you remember what happens, say for example, RNA synthesis has taken place. Now we are having two strands of DNA. One is the coding strand, another is the template strand. We have already talked about them. Okay, and the RNA, it takes the message of addition of nucleotides always from the template DNA, template strand, the template strand and the RNA formed, they are having the base pairing complementary. They are called, so the template strand is called as non-coding strand, and the RNA formed has the same sequence as that of the non-template strand, which has not participated in synthesis of RNA, and except for uracil, except for thymine, there will be uracil. This we have talked in previous lecture. You can go there. And we are having the transcription bubble. This is now ternary complex, open ternary complex. Why open? Because DNA has melted a bit, okay, and here we are having now three molecules of different chemical nature. One is RNA polymerase, an enzyme, another is DNA, another is RNA. So it will be now a ternary complex. See, once 8 to 9 nucleotides of RNA have been added, this sigma subunit, now its work is done. Its prime work was to recognize the promoter. It has recognized the promoter. Now its work is done. What happens to it? After adding 8 to 9 bases, it dissociates from the enzyme, and now we are left only with the coenzyme here. Another important thing about this RNA synthesis is that it's quite unstable. Unless and until we don't have 10 base pairs of RNA added, more than 10 base pairs of RNA added, we can't be sure that whether this RNA will elongate further or not. Up till 10 or lower than that, it's quite unstable structure and can abort. RNA synthesis can abort, which we call as abortive initiation. But once 10 base pairs have been added, we say a stable ternary complex is formed and the RNA synthesis is able to move forward. And another important thing about this RNA synthesis is that RNA polymerase does not require a primer. It's able to start the transcription de novo, that is, it adds first nucleotide itself and goes elongating it. Now after the initiation we say up to the stage up to 10 base pairs have been added. After 10 base and the sigma subunit has dissociated, now further it comes up under the elongation. Remember that it's single RNA polymerase which is responsible for transcription of all different types of RNA in prokaryotes. Also, as this RNA polymerase moves further along the DNA, it goes on unwinding the DNA duplex due to its helicase activity, and this unwinding provides the template for the synthesis of RNA. Now this RNA at the 3' end, we are having ribonucleotides added and the chain elongates. Also, as it moves further and this DNA, it has transcribed into RNA, now it rewinds back. And during the process of rewinding, many positive and negative supercoils are induced in it, means a wrong twisting happens in it, and luckily we are having the enzymes for that, and the job is done by the gyrase and topoisomerase first. Gyrase, it induces negative supercoils and positive supercoils are induced by the topoisomerase first. Now chemical mechanism in elongation. In nucleotide addition, in RNA, the beta and gamma phosphates are removed from the incoming nucleotide and the hydroxyl group is removed from the nucleotide present at the end of the chain. See what it means. We are having the growing RNA chain and this is the 3' end, 3' sugar OH group which is free and is ready to form bond with the incoming ribonucleotide, and we are having the ribonucleotide with a base with a ribose sugar and 3 phosphates. Now what happens, this oxygen, it makes a bond with the alpha phosphate here, and as a matter of that, these 2 phosphates, they are removed, that is beta and gamma phosphates, they are removed, which what we call as pyrophosphate, and also this O is removed from this and we are having the 3' 5' phosphodiester bond formed. Now coming to another, after the elongation is done, ribonucleotides have been added. Now comes the question of termination. That is when this RNA synthesis is to be terminated, is to be halted. It involves recognition of a point at which no nucleotides are added to the chain. Also, the enzyme finds a terminator sequence and stops transcription. Simply this one mechanism is simply this RNA polymerase, as it moves along the RNA, at some sequence there are hairpin-like structures which are palindromic in sequence, and the RNA polymerase recognizes them, and there the transcription is halted. No more nucleotides are added to this end. And after the termination has happened, we are having the disruption of the ternary complex, and we are having in cytoplasm, we are having DNA duplex which has rewinded, we are having an RNA polymerase, and we are having a newly synthesized RNA. This type of termination is called as intrinsic termination. Now coming to another termination, that is row dependent termination. In this simply a protein called as row, it recognizes a site in the RNA which we call as RUT site, R-U-T, site, row utilization site, and this row, it recognizes the site which is very rich in cytosine residue and poor in guanosine residues. It goes and binds there, and from here no nucleotides are added. That is the termination occurs. Hope this is clear. Now watch this video for more understanding.
From our diet. Our bodies cannot use the food as it is when it enters our digestive system. The process of chemical digestion uses different proteins and enzymes to break down the food particles into usable nutrients our cells can absorb. And where are the instructions to manufacture these and all the different types of proteins we need to stay alive? The instructions to make proteins are contained in our DNA. DNA contains genes. A gene is a continuous string of nucleotides containing a region that codes for an RNA molecule. This region begins with a promoter and ends in a terminator. Genes also contain regulatory sequences that can be found near the promoter or at a more distant location. For some genes, the encoded RNA is used to synthesize a protein in a process called gene expression. For these genes, expression can be divided into two processes: transcription and translation. In eukaryotic cells, transcription occurs in the nucleus, where DNA is used as a template to make messenger RNA. Then in translation, which occurs in the cytoplasm of the cell, the information contained in the messenger RNA is used to make a polypeptide. During transcription, the DNA in the gene is used as a template to make a messenger RNA strand with the help of the enzyme RNA polymerase. This process occurs in three stages: initiation, elongation, and termination. During initiation, the promoter region of the gene functions as a recognition site for RNA polymerase to bind. This is where the majority of gene expression is controlled, by either permitting or blocking access to this site by the RNA polymerase. Binding causes the DNA double helix to unwind and open. Then during elongation, the RNA polymerase slides along the template DNA strand. As the complementary bases pair up, the RNA polymerase links nucleotides to the 3' end of the growing RNA molecule. Once the RNA polymerase reaches the terminator portion of the gene, the messenger RNA transcript is complete, and the RNA polymerase, the DNA strand, and the messenger RNA transcript dissociate from each other. The strand of messenger RNA that is made during transcription includes regions called exons that code for a protein, and non-coding sections called introns. In order for the messenger RNA...
Well, this is all. Thank you.